And today I'm going to talk a little bit about my spring slice and the stretch effects on one of those, a couple of those. And so I don't know if in the room, in the audience, if people are familiar with the neuroscience, but here is typically what a neuron looks like. There's a cell body with dendritic trees. An axon would come out here that's wrapped by some other cells, and they form the sheets that actually insulate the electrical potential that goes through the axon. And it would go all the way to either a neuromuscular junction or neural junction, where a synapse would occur, which is over here. And so typically a neuron is characterized by two means. First, electrical potential. This is actually the first observed action potential back in 1939, and it earned Hawking and Huxley a Nobel Prize. And right at the synapse, it's mainly chemical. There's a lot of ion channels, a lot of metabolic pathways that regulates memory. In recent discoveries where there's super-resolution microscopy, people have actually found out that the axon is very organized in terms of its actin filament, microtubules, and the sodium channels. This is a very strong suggestion that mechanical simulations could also play a big role because everything is coupled, and when a certain mechanical stimuli is being inputted, it might lead to easier activation or whatnot. Here, what's spinning is a Drosophila embryo that's being dissected, and a single axon is isolated here. So our lab previously have actually bring in a MEMS device and probe what kind of tension is being maintained with one of these axons. It turns out it's in the order of 1 nanonewton. So typically what a fibroblast would exert on a PHL surface of 10 kilopascal. And interestingly, when you cut the axon, these vesicles that would accumulate here would actually gone. And when you pull it back, giving it back a mechanical tension, it will actually restore that clustering mechanism. And if you further pull it even more, then the vesicle clustering would go up by 200% by holding the axon for 30 minutes. And so, interesting. And there's also something else. Here on the right is a picture of actually, I believe, a couple of days of constantly stretching the neurons. And you can see how dynamic they are, that these thin fibers, in fact, the growth rate is around maximum, I believe, 8 millimeters a day, which is significant. And also, people have shown that in frog neuromuscular junctions, a stretch would actually enhance the release of neurotransmitter. It is endocrine dependent, for those of you who are interested in the mechanobiology. And going back to brain slices, so what has been done in the literature? So people usually study brain slices when it's traumatic conditions, and so they would induce some sort of blast, in this case a vacuum over here, and then it would suck in the membrane, which has some cells plated on it, and this way they would see the dynamics of the cells or culture or tissue. People have also invented some microelectrode arrays, which are flexible, and they would stick on top of the slice, and when it's being stretched, they would get the potential also. So all of that being said, how do I plan to apply a sort of these strains onto your brain? Because, as I've mentioned, in brain slice literature, mostly these are large strain and large strain rate, and they're supposed to be injury. But how do we actually study what we've talked about in these scenarios where a very small amount of force would lead to a difference in the neuroscience? And so this is the device that was made. This is the schematics. And we have two files. This is the slice, and this pathway is called the Callosal pathway. It bridges the left and right hemisphere. And so this slice is taken like this, coronal slice. ACSF would come in here, and then it brings out, and then this mechanism will actually stretch the slice uniaxially. This is a real live image here. And so we also are concerned about the strain measurements because ultimately we want to quantify what kind of things we're putting into the system. And so here is a map of displacement versus time. This scale is in microns. And so this device is able to give a resolution of around 10 microns. And it's pretty linear. And also by tracing the edges, you would be able to get the global strains that you put into the slice. And we also have another technique, which actually is mentioned last series by Dr. Han from Purdue. It's called digital image correlation. Pretty much you put two images together, you get the displacement field, and then if you know the material property, then you probably would get also the forces. This way we can also map the local strain in the region that we're interested in. And then, okay, we figure out the mechanical engineering part, which is easier. And then how are we going to get the neural responses? Because imagine, the slice is actually being stretched right now. So typically you would put an electrode in, but now it's stretching. So how do you synchronize the movement of the electrode, which turns out to be a huge challenge. There's also other imaging methods, such as calcium imaging and voltage-sensitive dye. These methods tend to decay over time, and they're not very stable. And you actually have to put in some extrinsic dye so that it would actually fluoresce under epifluorescent microscope. And so we came to this method, which one of our collaborators is a pioneer in. It's called flavored protein autofluorescence. And I would like to bring your attention down to here, number K. This is what people in the field think that is actually happening. So neural activities would lead to a spike in calcium. That's very well known. And then calcium increase would lead to energy metabolism in the mitochondria. They get hot, and then they oxidize some protein, flavor proteins, which gives you a signal. And so this is a plot where they match it to a field potential and also some calcium imaging. And the molecular basis is this. Back in high school biology, probably, we learned about Krebs cycle. So how glucose give you energy and give you ADP. And in fact, these two proteins are involved in the cycle. So whenever it undergoes metabolic pathway, these proteins will actually autofluoresce. So quite a contrary to Joanne, who doesn't want any autofluorescing. I do want them a lot. And what I image is the FAD, hence the name flavor protein autofluorescence. Okay, now we are able to image it. We could get some responses. So how do we actually capture what's activated, right? Each pixel, imagine we're taking a video of the slice when it's being imaged, right? And then every pixel will have an intensity versus time profile. So this is here. And you can get a standard deviation of that. So a higher standard deviation means that it's more active for obvious reasons. But how do you give it a threshold? So here is what we came up with. We fit it to a two-term Gaussian. Two-term because there's white and dark. And Gaussian means that it's random, right? So whatever is deviating from the random distribution tells you that they're actually actively participating. And if you zoom in really close to the right tail, which you would expect because of high activity, there you can establish a criterion where you can select those pixels. In F, this is a stack in image A where it gives you the standard deviation value, and then this is what was selected in MATLAB. So finally, we're ready for experiments after all of this, and I have 10 minutes left. So I would like to comment a little bit on the experimental paradigm first. So what we do is that we would do a baseline measurement first in the beginning. A baseline measurement means that the slice is mounted on the device. The camera everything is ready. And then we would give it an electrical, in-field electrical potential characterized by this profile. It's every 10 seconds, and then you have some amplitude every 40 milliseconds. And we would try to get it down to around 20% probability of firing. And once this is achieved, then we will stretch it and hold it for 10 minutes, no imaging whatsoever. And then we bring it back, and then we do the imaging again. So theoretically and experimentally, the conditions for baseline number 1 and baseline number 2 should be the same. And if the stretch has no participation in the process, you would expect no changes. And this is repeated for 5 times for each experiment. Here is the data. In the black bars, this is the stretch enhanced group, so we undergo the stretching manipulations. The control experiments is just that the slice is mount, and you keep on stimulating it and imaging it without undergoing through the stretch manipulations. And so you can clearly see that there's a difference, which you would not expect if mechanical stimuli has no participations. And then the plot on B is just showing that if I take every experiment and I give it, assume that it's linear and then give it a slope, the stretch enhanced group would have a slope of 0.09, whatever that means. However, the control group is pretty flat. And this is statistically significant. And this is a little bit overwhelming. But so remember, now I'm able to select the active pixels with the criterion that I've established. And so I can actually do some area analysis because I can select the pixels. And turns out that in the control case, the activation area is plateauing, very flat. And in the stretch enhanced case, it would actually go up and down. B is just a map to show that initially, the flavor protein autofluorescence, which we call FA usually, the FA response is very similar. But as it goes to higher and higher stretching manipulations, this amplitude will actually go down. This is what the plot here is showing. Black is again the stretch enhanced group. Control is remain relatively stable. Other than that, we can also get the rates of activation by measuring the slope here. And so this is what we're trying to show here in D and E. One is the amplitude, average amplitude of all these cases. And the slope of it versus the baseline number. And in the control case, and here, apparently, the flavor protein, the FA response isn't as strong as you would see in control. And I will come back to that, trying to explain why this is happening. And in F and G, so in D and E, these are only baseline number that we're comparing to. And here we're trying to actually, because we know that when you stretch it, it's actually more excitable. And so if we try to do it with electricity other than mechanical stimuli, would we be expecting the same decrease in the FA potential, FA amplitude and the rate? And so this is not the case. So we can, this is verifying that this difference is caused by the stretching manipulation, but not the excitability. And this one is a little bit different. So in these sets of experiments, what I did is I would image the slice, and then I would stretch it. I would image it while I stretch it. And these two plots, the red and the green, shows the two strain level that I mentioned previously, the global strain and the local strain from DIC and tracing the edge. As you can see, the response corresponds very well to some of these actual stretching manipulations. The blue line is the FA response, and these are the corresponding area map. And this is telling us that the slice is sensitive to stretch, a static stretch, but it's also sensitive to a stretch impulse. And the response is instantaneous. 50% of the time that I have this, and you can see some of these cases it didn't show up. Okay. Oops. And so this is my second last slide. So what I would like to conclude is that this is the first attempt to actually have a brain slice being able to mount a device and put under a microscope that you can actively image it. Some hyperexcitability is observed, and I would like to comment on what we mean by hyperexcitability here. So back in this plot, you can see that the excitability is going up on the y-axis. However, the FA amplitude and the rate is going down. And we think that it's because now whatever is limiting this FA signal, the activation potential is actually lower. And hence, it's easier to activate. That's why the high excitability and the lower amplitude and rate, because it's easier to excite. But what we know is observational, and we don't really know what's underlying all these mechanisms. And so what we plan to do in the future is to do some pharmaceutical studies, let's say using blebbistatin to target actin to see if we still see it. We can do some knockdowns with knockdown mice. And we would like to see if this phenomenon is repeatable in a cellular level. Although I mentioned that calcium imaging isn't stable as compared to FA imaging, it gives you a very good cellular resolution. And there's these mice these days called GCAM, where it's genetically modified to have calcium fluorescing rather than you having a Fourier dye to dye the tissue. That's it, and I would like to thank my collaborators, my PI, Professor Saif, Daniel Liano from Beckman, his grad student who've done all the experiments with me, Kevin, BJ who've helped, Rainer who critiqued, and my group here and the funding agency. So I would like to open the floor for questions.
Anthony, can you just repeat the questions that anybody asked you so that everybody at UC Merced can hear them?
Okay.
I guess they can hear them.
Hopefully there are questions. Go ahead. Sorry.
Actually, I have two questions. One is like, do you keep it in media and temperature and CO2? And second is, how do you deal with hysteresis?
Hysteresis meaning?
It's like you stretch and you put it back and you stretch again, right?
Mm-hmm.
So like any mechanical material, they always like hysteresis.
Yes, the hysteresis actually is in the energy. However, the position remains the same. So if you plot the stress-strain curve, I don't know if you learned about it, you would initially get a slope, and then if it plastically yield, it would go plateau, and then it would come back down, and then it would go back to the exact location. What's the hysteresis mean is that this curve actually has an area, and there is an energy loss. However, the position could be maintained. And the first question is, yes, we perfuse the slice with artificial cerebral spinal fluid. It's pretty standard in brain slice experiment. And we keep the temperature 28 degrees C. This part, people also always get questioned. Because we keep it at 28 degrees C because the FA signal is better at that temperature. We would like to do it in 37 degrees C for obvious reason because it's, this is the in vivo condition. But due to technical limitations, we aren't able to do that. Yeah.
So you think that this temperature difference would introduce some changes as compared to the normal physiological condition?
Right. That's a very good question, and I have no good answers to it because I have not done it yet. However, as I mentioned, the FA signal gets tendered very quickly as the temperature goes. So we'll have to resort to another method to answer the question. However, I would assume so, because our leading hypothesis for this phenomenon now is based on vesicle clusterings. So these things cluster first with active transport, but also with Brownian motions, and Brownian motion has a very heavy dependence on temperature. But it's not that much different, so I don't know for sure.
Was there a reason you held the stretch for 10 minutes?
Yeah. So, I don't know if you remember, in the PNAS work, in the stretching neuron and Drosophila work, the stretch was held 30 minutes there. So we want to match that time scale. But here, because we want to utilize the whole lifespan of the slice, which is roughly 8 to 10 hours. And so setting it up is already some time, and we would like to do the imaging as short as we can so that it doesn't get filled with bleach and all those stuff. Autofluorescence also get filled with bleach. And so 10 minutes is really the thing that we think would fit everything together.
Did you try any other different time points?
We plan to. Yeah.
Okay.
Go ahead, Brian.
So you mentioned with the longer stretching up to 30 minutes, then you observe the more clustering of the vesicles at the synapse. With the 10-minute stretch, have you looked at the vesicle clustering?
Yeah, it's a lot difficult, much more difficult in brain slices, if I am correct, because I think the only way that you can visualize vesicles effectively is by doing electron micrographs, and it's extremely difficult to do. We do plan to do some, hopefully to see if there actually is in one of our proposals that we want to try electron micrograph to see directly visualize the vesicles. You can also tag them, but I think the signal is not very good because it's a tissue. Whether in Drosophila, it's really like it's cells, and you can lay them down flat.
And this is a very basic question. How do you exactly measure the degree of excitability? Is that just the probability of firing?
Right, the probability of firing. So in the initial setup, I always tune it down to around 20% of firing, and then it goes up. You would see 30%, 50% for every electrical stimuli. So for every 5, on average, for every 5 electrical stimuli, if you get 1 response, then that's 20%. Am I making sense?
Okay. So you're giving the electrical stimulation.
Right,
while-
correct.
Any more questions? When you put these samples on there, can you use these samples later for other analyses if you want to do like any bulk analyses with the samples to see if there's any changes between different groups?
Right. So usually people just toss them away, but if you fix them, I think you can do a lot of histological work.
More questions? All right, let's thank Anthony.
Thank you.